Dear friends and colleagues, first of all, Happy New Year. Welcome to the 2026 new series of APSHG Hong Kong GI Distinguished Scholar Series lectures. My name is Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute and also the immediate past president of the Asia Pacific Society of Human Genetics. It gives me great pleasure to introduce our lecturer, our scholar, for today's Scholar Series, Professor Raju Kuchalapati. Professor Kuchalapati is the Paul Caput Professor of Genetics and Professors of Medicine at Harvard Medical School and was the first scientific director of the Harvard Medical School Partners Healthcare Center for Genetics and Genomics. Professor Kuchalapati contributed to several different areas of research in genetics and genomics in human cancers. His team is a part of the Human Genome Program and the Cancer Genome Atlas Program. He served on the editorial board of the New England Journal of Medicine and was editor-in-chief of the journal Genomics. He's a fellow of the American Association for the Advancements of Science and a member of the National Academy of Medicine. Professor Kuchalapati was a member of the Presidential Commission for the Study of Bioethical Issues. He also serves as an advisor to the Center for Medicare and Medicaid Services for reimbursement of laboratory-developed tests. The title of his talk for this Hong Kong GI-APSHG Distinguished Scholar Series is Impact of Artificial Intelligence on Cancer Precision Medicine. Please enjoy.
Thank you for inviting me to make this presentation. Today I would like to talk about the impact of artificial intelligence on cancer precision medicine. The care of cancer patients has been evolving significantly over the last century or more. At the beginning of the last century, the only tools that were available to treat patients is to remove the tumor through surgical means. And surgery continues to be a very important way of treating cancer patients. Indeed, in some countries, such as China, surgeons play a very important role, not only removing tumors, but actually taking care of them after surgery. Also, at the beginning of the last century, the recognition that radiation is capable of killing tumor cells provided a new opportunity to treat patients. And radiation therapy continues to be a very important mode of treating cancer patients, together with surgery and sometimes radiation therapy alone. In the middle of the last century, it was recognized that certain types of chemical agents are capable of killing cells that are growing rapidly. And since the cancer cells in general have a tendency to grow rapidly, it was thought that it is possible to treat patients with these chemotherapeutic agents. Many chemotherapeutic agents have been identified, and chemotherapeutic agents singly or in combination have been and continues to be an important way in which we treat cancer patients. An additional set of tools really became available at the beginning of this century, and this is now enabling us to provide a different way of treating patients, which is called precision medicine or personalized medicine in cancer treatment. And I'll tell you more about both of these aspects. The precision medicine really began with the sequencing of the human genome, which started in 1990. And the initial phase of this human genome sequencing was completed in the year 2000. And a couple of magazines, Nature and Science, devoted their entire issues to the descriptions of the human genome in 2001. So it took 10 years to sequence a single human genome. And as I've indicated, it was both a public effort that was initiated initially in the United States, and later on other countries, including China, has joined these efforts, and therefore it became an international effort. And thousands of people were involved in sequencing the first human genome. And I had the privilege of being a part of mapping and sequencing the initial human genome. And it was estimated that the cost of sequencing a single human genome at the time was $2.5 billion US. Since in the last 25 years, there have been very significant changes in the sequencing technologies. And on the upper left-hand corner of the slide, you could see the way that sequencing was done. Many DNA sequencers were set up to be able to sequence the human genome. And because the capacity of each of these machines was not very significant. And things have really changed now in the last 45 years. And many companies, such as Illumina in the United States, developed the machines that are shown on the upper left-hand corner. And the Beijing Genomics in China has developed sequencing tools and machines that are shown on the upper right-hand corner. And a British company developed new kinds of technologies to be able to do in situ sequencing that are shown on the bottom right-hand corner. The technology of sequencing has evolved, but the cost of sequencing the human genome has also been reduced very significantly. For example, one of the laboratories in Cambridge, called the Broad Institute, has a clinical laboratory, and this laboratory is capable of providing short-read whole genome sequencing on the Illumina machine, and for a cost of less than $400. And Illumina and other companies have recently made announcements that one of those companies, Ultima Genomics, last year made an announcement that they would be able to provide sequencing for less than $100. I had the opportunity to visit some of the new companies in China, and these companies are developing new technologies to provide genome sequencing for much less cost, and it might be possible to do sequencing for a few hundred RMB, enabling us to be able to sequence larger numbers of genomes when it is necessary. The ability to sequence the human genomes really enabled us to think about precision medicine or personalized medicine. And 10 years ago, President Obama in the United States recognized the importance of precision medicine, and he defines this precision medicine as delivering the right treatments at the right time, every time, to the right person. And since these 10 years of declaration of the importance of precision medicine, cancer care has significantly improved as a result of all of these new technologies. And today, in the 2020s, artificial intelligence is impacting very large numbers of our human enterprise, and artificial intelligence is also helping advance precision medicine. I'll give you several examples of how precision medicine and artificial intelligence are coming together. And one of the greatest hopes of precision medicine is that we might be able to conduct early detection of cancer and intervene early. Because there is significant amount of evidence that indicates that if you are able to identify cancer early, that the chances that those people would be able to survive for 5 years or more is 95%. Whereas if you would diagnose cancer late, like stage 4 cancer, the chances for their long-term survival is less than 5%. So early intervention would be very helpful. And there are many different ways in which you'd be able to treat and prevent cancer. And one of them is that many cancers are known to be the results of infection by viruses. For example, Epstein-Barr virus is capable of causing Burkitt's lymphoma, Hodgkin's disease, and nasopharyngeal carcinoma. Papillomaviruses 16 and 18 are capable of causing cervical carcinoma and other cancers. And Kaposi sarcoma is also the result of a treatment associated with herpes. And infections with hepatitis B and hepatitis C, some of them are very common in China, are known to cause hepatocellular carcinoma. There are many other cancers that are caused by viruses, and there are vaccines against several of these viruses, and administrations of these vaccines are indeed capable of preventing cancers. The dramatic cases of them are vaccination against papillomaviruses in 1618, and as a result of that, in some parts of the world, these immunizations were given to young women, and these vaccinations are capable of preventing and reducing the incidence of several carcinoma greatly. So all of these attacks of virally induced cancers can be prevented using this approach. Now, in the cases there are some cases in which cancers are known to be run in families, and those types of cancers are considered to be the result of a cancer predisposition. Best examples of a cancer predisposition are breast cancer and colon cancer. In the case of breast cancer, children and that are born with mutations in BRCA1 and BRCA2, as they become older and become women, they have a tendency to develop breast and ovarian cancer before they reach age 50. And it also turns out that sporadic cancers, or those cancers that do not have a family history, also have, some of them are the results of mutations that the patients are born with, or germline mutations. And it turns out that 10% of all cancers are the result of genetic predisposition. So if these cancers are the results of a genetic predisposition, then sequencing the DNA from these individuals, even before they develop the cancer, we would be able to identify the susceptibility. And in many cases, therefore, you'd be able to utilize that information to either prevent cancer or to start treating them early in their cancer development. So it is now recommended in the United States that all colorectal cancer patients should be tested for genetic mutations that cause a disease called the Lynch syndrome. So now it is indeed possible, using these new DNA sequencing technologies, to sequence the genomes of all the people who are considered to be at risk, and you would be able to identify those individuals who carry germline mutations that predispose them to varieties of different cancers, so therefore we'd be able to treat them effectively. It turns out that new methodologies are becoming available that use artificial intelligence to detect cancers early. In these examples, a number of images that are taken from patients who were diagnosed with breast cancer have been examined, and a tool has been developed by my colleagues here in Boston that is headed by Regina Barzilay from MIT, and she and her colleagues developed this tool called Sybil, which is a validated deep learning model, predict both lung cancer and breast cancer. And this program is capable of detecting cancers effectively, but it turns out that in some cases it's also capable of detecting tumors, even before the patients have clinical symptoms. And the examples of them are shown on the right-hand side. So you could see in the figure A that there is a lesion that could not under normal circumstances be considered to be a tumor lesion, but that was recognized by Sybil as a possible tumor lesion. And so that patient was followed, and 2 years later you could see with the arrow that indeed it really turned out to be a tumor. Similarly, in the case of C, it's an early diagnosis that showed a small tumor or lesion that turns out to be a true tumor lesion as it goes and in image D. So this kind of a technology then therefore be used not only be able to detect the tumors effectively using these imaging technologies, but it is also possible to identify tumors even before the patients develop clinical symptoms. This is an example of a lung cancer, and this also turns out to be true in the case of breast cancer. Here is the situation on the left-hand side when this program was able to identify a lesion in the breast that the machine considered to be a tumor. But this individual did not have any clinical indications of the presence of the tumor. And so this patient was followed carefully. And 2 years later, the same imaging technology now reveals a true tumor lesion and will indicate that, that this tool is very effective in being able to do that. And so these types of technologies are now being implemented routinely in imaging the tumors, and bringing these artificial intelligence tools to medical imaging is changing the way that we are able to do medical imaging. First, use of these artificial intelligence systems are changing the time that is required to be able to acquire the tumors, and therefore the patients don't have to be exposed to radiation for long periods of time. And the workflow can be automated. And in the case of oncology, as I've shown you, it is possible to use these methodologies to detect automatically the tumors. And it's also possible to use these methodologies to measure tumor burden or use other types of technologies. These types of beginning to bring AI and medical imaging are not only helping cancer, but it also is helping many other fields of medicine, including neurobiology, cardiac biology, and metabolic assessments. So this is going to have a revolutionary impact on the way that imaging and artificial intelligence are going to come together. It is now possible also to predict cancer risk just based on the clinical data and the use of artificial intelligence systems. This is a paper that is published, was headed by my colleague Chris Sander at Harvard Medical School. And Chris and his colleagues used clinical data from a large number of patients initially from Denmark, but later from the United States, and to try to identify symptoms that may be present from clinical data if they are going to develop cancers. In this particular case, they focused their efforts on pancreatic cancer, and they were able to show that just using clinical data without any special methods to use, that it is possible to be able to use these error minimized neural network systems to be able to identify the people at risk. And so the overall population can be divided then into those people that are at higher risk, and those people at higher risk can then be, monitored or surveyed to see if they would be developing cancer. So you would be able to identify those cancers, very early. So even though this work is initially done with pancreatic cancer, efforts are underway to try to use these types of technologies to look at other types of cancers and whether we could identify people at risk using clinical data. It's also possible to identify biomarkers that could be present in the blood of patients that could be used for early detection. Here's an example of some of the work that we have done in collaboration with Sam Hanash and Harold Warmus and other colleagues. And in this particular case, we developed a number of mouse models that spontaneously develop lung tumors. And each of these different models that are shown on the bottom left-hand side, the tumors were analyzed and the plasma samples from these tumor-bearing mice, and before they actually developed the tumors, were examined by for proteomic analysis using mass spectrometry. Using these methodologies, it was possible to identify a number of different genes and gene products that can be identified prior to these animals developing tumors. So these markers are being tested in human patients to see if it is possible to use them. And indeed, there is evidence to suggest that some of these markers that have been identified in the mouse are also useful to identify human tumors. We have done another type of experiment to try to identify markers before the patients were diagnosed with cancer. This study was facilitated by the so-called longitudinal cohorts. In this case, we used the cohort that started in 1993, which is called the Women's Health Study, in which at that time nearly 40,000 women were enrolled in this program in 1993. And every 2 years, they provide all of the clinical information. They also provide blood samples, and from the blood samples, the Buffy code and the plasma were separated and stored for future analysis. So if a patient were diagnosed with a particular cancer that is shown by the arrow, that means that that patient has provided a blood sample prior to the diagnosis of cancer. So it is possible to be able to take those samples prior to the diagnosis and examine those plasma samples by a mass spectrometry. And when we did that, we were able to identify a number of different markers that were indeed present prior to diagnosis of the cancer. Therefore, we could use those types of cancer biomarkers to identify cancers early or even before cancer develops. The human genome sequencing and the precision medicine is also enabling us to think about, caring for cancer patients who are diagnosed with cancer. To understand more about this cancer, a program was started in the United States, and again, I had the privilege of being a part of this program called the Cancer Genome Atlas. And this program was started in 2006. And in this particular case, as shown on the left-hand side, a large number of tumor types were identified for analysis. And in each of these cases, we obtained 500 tumor samples and also obtained blood from those patients with the tumors, and the blood served as a normal control. And the tumor samples and the blood samples, we extracted DNA, RNA, and protein and subjected them to genetic and genomic analysis. And that program, as I mentioned to you, that started in 2006 and was completed around 2018, resulted in a large number of publications that described the genetic and genomic features of all of these different cancer types. I'll give you examples of the kinds of results that are obtained from those studies. Here's an example of the results that we obtained from sequencing the DNA from several hundred samples from patients who've been identified with non-small cell lung carcinoma. And these are the results that are obtained by sequencing all of the 20,000 genes in the tumors and the normal samples. And the results are summarized in this figure. There are a number of interesting aspects that can be gleaned from these results. First, even though 20,000 genes have been sequenced, as you can see on the left-hand side, only about 20 of these 20,000 genes are ever found to be mutated in these tumors. And some genes, such as the one that is shown at the top, TP53 gene that is most frequently mutated, 46% of the tumors had such mutations. KRAS was the second most frequent mutation, with 33% of the tumors having mutations, and so on and so forth. The other interesting feature is that if you examine any one individual tumor, of these 20 genes, there are only at most about 4 or 5 genes that are ever found to be mutated in a given tumor. And so that means that these tumors are the results of these types of genetic changes, and each tumor can be characterized on the basis of the specific types of genetic changes that are present in that tumor. So we asked whether these types of changes are specific for non-small cell lung cancer or whether it is true for other types of cancers. And it turns out that the same types of results are true in virtually all of the cancers that we have examined. On the left-hand side, you could see again the results from non-small cell lung cancer. In the middle panel, you'd be able to see results from pancreatic cancer, and on the right-hand side, be able to see the results from bladder cancer. Again, as you see in each of these cases, only about 20 or 25 of the all the 20,000 genes are found to be mutated. In each tumor type, the relative frequency of each of these genes might be different. And as you could see, in non-small cell lung cancer and in bladder cancer, TP53 is the most frequently mutated gene. In the case of pancreatic cancer, KRAS is the most frequently mutated cancer. And so only a subset of genes then are specific for each of these cancer types. So one could look at all of these different cancer types, and they can be categorized into uh, uh, the driver genes. And it turns out that these driver genes are either tumor suppressor genes, where both copies of the gene have to be altered, or oncogenes, where a single gene alteration would be sufficient to drive the cancer. And but what is also interesting is that our ability to identify all of these different mutations in these different types of cancers enabled us, the pharmaceutical companies, to be able to develop drugs that are specific for each of these different genetic mutations that are present. And as shown on the right-hand side, it now turns out that there are varieties of different drugs that are available to be able to treat each of these different types of cancers. And some of them have already been approved by the Food and Drug Administration in the United States and by PMDA in China and other regulatory agencies throughout the world. Other drugs are in clinical trials, and yet others are in preclinical studies. In addition to identifying all of these cancer genes, it is also possible to find out where these mutations are within and how they affect the protein structure or protein function. And here's an example of the types of mutations that have been observed by the Cancer Genome Atlas in a gene called BRAF. And all of the different mutations that have been found out has been shown. And one of the most frequent mutations is the one that is shown on the right-hand side, so V600E. And you could exactly see where how these mutations affect the structure. And that means that by having this knowledge, that it is possible to be able to try to develop drugs. And I'll talk more about that a little bit later. As a result of all of this information and the efforts by pharmaceutical companies and biotech companies, the number of drugs that are available and how you treat the patients have really changed very dramatically. And here's a paper that was published in 2004 in which the lung cancers, non-small cell lung cancer patients, have been classified on the basis of the types of mutations that are present. And you could see that there are interesting observations here, is that although the patients from Asia and Caucasian patients, the genes that are mutated are similar, the relative frequencies with which those genes have been mutated are different. For example, in the case of Asians, epidermal growth factor or EGFR mutations are present in 35% of the Asian population, and sometimes as high as 20% in the Chinese populations, whereas in Caucasians, those mutations are present only in about 20% of the patients. Similarly, KRAS mutations are present only in about 3% of the Asian populations, whereas KRAS mutations are present in more than 15% of the patients in Caucasian populations. So this type of information is very important in not only classifying these tumors on the basis of these, but this information can also be used to be able to treat patients. And how this information is changing the way they will be able to take care of patients is shown in this slide. On the left-hand side, we're talking about all of the driver genes that are important for non-small cell lung cancer. And in 2001, 25 years ago, there were no drugs that targeted any of those mutations. 10 years to 50 years later, in 2014, there were a number of different drugs that target epidermal growth factor and a translocation called ALK that were available. 5-6 years ago, in 2019, there were more than 20 different drugs that were available for a variety of different mutations that are present in these non-small cell lung cancer tumors. In 2025, there are more than 100 different drugs that are available to treat patients. So as a result of the availability of all these drugs and our ability to be able to classify these patients based upon their genetic changes, there have been dramatic improvements in outcomes for these patients. At the beginning of this century, if a patient was diagnosed with non-small cell lung cancer, the long-term survival for those patients was poor. Today, in 2025, if a patient was diagnosed with non-small cell lung cancer, even at late stages, the long-term survival is very, very good. And 10 to 15% of the patients are now living for more than 5 years. And if a patient lives for more than 5 years, that patient is considered to be cured of the cancer. So you could see how dramatically genetics and genomics is improving the quality of cancer patients. Genome sequencing provides other types of information besides genetic and genomic changes, and that includes changes in copy number or copy number variation. And for example, it turns out that certain breast tumors, the gene HER2 copy number increases or is amplified, and those patients respond very well with HER2 antibodies. There are some patients in colon cancer, other types of cancer, where epidermal growth factor gene is amplified, and in yet other genes, the oncogene MET is amplified. And these amplifications also provide information about how to treat these patients. It's also possible to look at the RNA from these tumors and generate expression profiles and measure the levels of RNA corresponding to each of these genes. And based upon that, we can identify genes whose are overexpressed or underexpressed in tumors. For example, in the case of breast cancer, it turns out that the HER2 gene is overexpressed, and those tumors that have high levels of HER2 expression are ideal candidates for treatment with Herceptin. A new type of change, which is called the genetic rearrangement, where genes that may be located in different parts of the genome are connected to each other or translocated to each other or undergo gene rearrangement. Examples of done in the non-small cell lung cancer are genes called ALK, RET, and ROS, and these genes are not active in normal lung cells, but in these tumors where these translocations occur, they become activated, and there are now drugs that are available to treat these activated genes, providing new ways of treating patients. Finally, the types of genetic changes that I mentioned to you can also be used to determine what that gene is and what pathway that gene works on. And if a gene works on in a particular pathway, then it is possible to be able to develop drugs that treat the activation of that pathway. For example, there are a number of genes in the RAS MAP kinase pathway, such as EGFR, RAS, MAC, and other genes. And so if any of these genes are activated, that pathway is turned on. And so people have been trying to develop drugs that would inhibit the pathway. There would be a new way of treating these cancer patients. In addition to looking at these genetic and genomic changes, it's also possible to measure the number of mutations that are present in any given tumor. And the number of mutations that are present in a tumor is referred to as the mutation burden. And in the figure that is shown on the left-hand side, each dot represents a single tumor, and the number of somatic mutations that are present in the tumor is indicated by the on the on the y-axis. And as you can see, the certain types of tumors, like the very ones on the left-hand side, like pleocytic astrocytoma or acute lymphocytic leukemia or medulloblastoma, the tumors in general have low frequencies of tumor mutation burden, whereas on the extreme right-hand side, tumors such as melanoma or lung squamous cell carcinoma or lung adenocarcinoma in general have a higher tumor mutation burden. So a number of studies have been done to try to determine the causes for this high mutation burden. And it turns out there are a number of different reasons why tumors have high tumor mutation. For example, the reason why melanomas, which are on the extreme right-hand side, have very high mutations is because these are skin tumors, and skin is exposed to ultraviolet light, and ultraviolet light, causes genetic mutations in cells that could cause tumors, and that's the reason why those tumors have a very high mutation burden. Squamous cell lung carcinoma are highly frequent in individuals who have a smoking history, and smoking causes a mutation, and that is the reason why squamous cell lung tumors have high mutation burden. Other types of tumors, such as colorectal cancer, have high mutation burden, and some of these tumors are known to have mutations in genes that involve so-called microsatellite instability, and those tumors that have this microsatellite instability have these high mutation frequencies. So there are many different types of inducers that are capable of providing these types of mutation burden data. And as I'll show you in a moment, it turns out that the knowledge of mutation burden is also very important in how you might be able to treat patients. So once you have all of this information, it is important to be able to use all that information that is shown in the middle here, the structural variation, the mutation information, genetic expression variation, copy number variation, methylation changes in genomes. All of this information can be collected from these genomic types of analyses, and all of those data sets, which are different, have to be integrated, kept in a database, and annotated. And in tumor patients, once you obtain this genomic information, it is necessary to be able to compare the information from the individual patient with all of the data that are present in databases and be able to generate a report that could be sent to the caregivers to give them recommendations about treatment possibilities for each of these patients. Such a genetic testing is now available through many commercial agents, and it turns out that gene testing panels have been described, and on the left-hand side is a gene testing panel that has been described by University of California in San Francisco, and a total of about 600 genes of the 20,000 genes in the human genome are ever found to be mutated in all of the cancer types. So if you, in a single test, it is possible to sequence all of these 600 genes and be able to determine which genes are modified and use that information to make decisions. Now, many companies, Foundation Medicine and Grail and other companies such as Genetron in China, all of them provide these types of genetics and genomic tests. So as I pointed out, that even though the number of genetic changes in a tumor are relatively small, the overall data that is generated, from all of these patients is very, very significant. And the data sets from these changes is expanding and is coming from many different sources. And to save the cost of sequencing and provide this testing in a clinical setting, it is now the current general practice to sequence only the tumor DNA. And that means that once you sequence the human DNA, you have to remove all of the common variation that is present among human populations. And so that means you need to compare them with normal DNA samples. And there are already databases of DNA sequences from normal individuals. And so the normal and tumor samples have to be compared with each other. And new papers describing the effects of variants or genes and how they affect the biology of the tumor are also coming together, and we need to keep track of all of this. And the only way that you'll be able to do all of these different things is the use of artificial intelligence systems. So these artificial intelligence systems are starting with large language models that can be used to look at papers that are published in the literature and identify those that are important and use natural language processing methods to identify the critical information from those patients, and that can be used for deep learning and machine learning, and ultimately that will lead to artificial intelligence systems which are either supervised or unsupervised. That will really help in keeping track of all of this information and making it simpler and faster for genetic and genomic analysis. It turns out that all of these types of genomic analysis are also useful to develop new types of treatments for patients. Here's an example of one of those. And these mutations that I described to you earlier, then some of those mutations are actually result in amino acid changes. And some of those amino acid changes are non-conservative amino acid changes, and that gives rise to novel peptides. And those novel peptides are now recognized by the human body as a new antigens or recognized as neoantigens. And these tumor cells that are producing these new antigens are capable of presenting them to T cells. And under normal circumstances, it turns out that the T cells are prevented from attacking those tumor cells. But there are new drugs that are actually capable of removing that inhibition and T cells to kill the tumor cells. And in other cases, that the tumor cells that die and their peptides can be go to the antigen presenting cells, and they can stimulate, these antigen presenting cells can stimulate T cells and B cells to kill the cells or produce new antibodies. So one of these new areas that is developing as a result of that is the cancer vaccine development. And based upon these new peptides, that is possible to be able to have either cell-based vaccines or viral-based cancer vaccines or peptides. Neoantigens can be used as peptide-based vaccines or nucleic acid-based cancer vaccines. And that new methodologies, by looking at these types of changes, that it's possible to be able to determine which B cell epitopes or T cell epitopes can be generated using these antigenic peptides. And it's expected that these types of vaccines are going to play a very important role in how we'd be able to treat cancer patients in the future. AI is also playing a very important role in treatment strategies. And this is a paper that is published by a British group that had developed mathematical models for deep learning that enables adaptive personalized therapy. And these computerized methodologies have been compared with uh, uh, other methods. And as you could see on the right-hand side, the dashed line represents the outcomes in the clinic. And these, mathematical model-driven strategies, which are in all cases are either as good as the ones that are currently used, or in some cases much better outcomes for these patients. As I mentioned to you earlier, there are now new types of drugs called immune checkpoint inhibitors. These are capable of harnessing the body's own immune systems, and several drugs, that are capable of conducting these immune checkpoint inhibitors are now available to treat them. And it turns out that, however, not all of the patients are respond to these immune checkpoint inhibitors, and it is now possible to identify which of the patients are capable of responding to these immune checkpoint inhibitors. Here's an example of that where RNA sequencing enables one to identify markers of immune response. And when you know these markers, that you'd be able to predict the immune therapy response. So the combination of the use of mutation burden and these types of new models using expression-based profiles or changing the way that would be able to identify patients who are highly likely to respond to tumors and those that cannot. Artificial intelligence systems are also changing the way that we'd be able to develop drugs. And this is a company that was started by Google in the United Kingdom, and has a deep mind that has solved the 3D structures of virtually all of the human proteins. And the availability of these three-dimensional structures can now help us identify new small molecules that are capable of binding to specific regions of molecules. Here are some examples of molecules in G protein coupled receptor proteins. And so one could predict whether or not a molecule would be capable of inhibiting the function of the proteins or augmenting the functions of these proteins. As a result of these types of technologies, now artificial intelligence systems is playing a very important role in drug development. Now, instead of having real chemical libraries, now there are virtual libraries that contain billions of compounds, and using these artificial intelligence systems is possible to screen these billions of compound libraries against the particular target that we're interested in, and reduce those complex from a billion compounds to a few hundred. And then one could then pick the most appropriate ones for further development. These types of changes and other types of changes in drug development are having a very significant impact in the way that drugs are going to be built. And to summarize, it is possible now to identify individuals at risk for developing diseases, including cancer. We have early detection markers that could identify people's very early, or in some cases even before they develop cancer. For those patients who have been diagnosed with cancer, it is possible to use genetic and genomic information for making clinical decisions. And these large databases that are coming out from all of this data are increasing in size, and artificial intelligence systems are helping in treating all of these patients using these new methodologies. So it's already having a very significant impact, the precision medicine and AI, and in the future, this continues to transform the way that we'll be able to take care of cancer patients. Thank you very much for your attention.